welcome Dr. Ahmad Fathoni, M.Eng, Head of the Center for Applied Microbiology BRIN. Our speaker, Professor Dr. Wong from Universiti Putra, Malaysia, visiting researcher of Research Center for Applied Microbiology BRIN. And our moderator for today, Dr. Neo Endra Lelana, researcher from Research Center for Applied Microbiology BRIN. Ladies and gentlemen, next agenda is singing the national anthem, Indonesia Raya. Ladies and gentlemen, next is opening remark from Head of Research Center for Applied Microbiology BRIN, to Dr. Ahmad Fathoni, time is yours.
Oke, cek tes. Udah bisa, Mbak? Halo, halo.
Clear, Pak.
Ya, suara saya jelas ya? Clear ya?
Jelas, Pak.
Oke, Bismillahirrahmanirrahim. Honorable Speaker, Prof. Wong of Universiti Putra, Malaysia. It's really nice to see you again this afternoon. And Bapak Ibu sekalian, all participants, Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. First of all, I would like to thank Prof. Wong. She's here actually with us in BRIN in Cibinong as visiting professor, and it is a great honor to have Prof. Wong, who will talk about a plant disease management. So she will share her experience, long experience during her work in Malaysia, and hopefully during her stay here 1 month, yeah, Prof. Wong, yeah. So yeah, so she will stay here for 1 month as a visiting professor, and hopefully we can gain more knowledge, and then share information and share experiences. So please, everyone, she's based in Cibinong, so anytime, yeah, please drop by to laboratory in bacteriology laboratory, and I'm sure she will be very welcome to have a discuss, and also even on potential future collaboration with us, BRIN and UPM. Yeah, and I would like to also thank you everyone for coming and then joining our webinar this afternoon. Yeah, actually this morning I tried to connect at 9 in the morning, and then I just got wrong schedule. So because usually we have seminar in the morning, Prof. Wong, yeah, 9 in the morning, but yeah, this morning I click Zoom at 9, and then only me, and then I check the schedule. Oh, it's in the afternoon, 1:30. I got wrong schedule. So, but okay. Yeah, everyone, thank you very much again for your coming. And as we know that the climate change has affected much, yeah, in our life, in many ways, especially in agriculture sector, yeah. And then, yeah, climate change cause drought, and then heat stress, and then also reduce the agriculture productivity in a way. One of its increasing plant disease. This is also one of, I think, due to the climate change. Yeah, so we may notice the increase in plant disease event, and also, more disease also come up, yeah, due to the climate change. And then disease also can have mutation due to the climate change, and even make us more difficult to tackle or to overcome the plant disease. And plant disease is one of the key, I think, the key aspect, or key factor that influence our crops productivity. That's why before we can handle them, we need to be able to identify and then know what they are, whether they are new or they are already existing. And then, yeah, and then there are several approaches that we can do also to tackle the plant disease. But here this afternoon we will hear Prof. Wong will share her expertise on how we develop, yeah, how she developed plant disease management through molecular approach. And hopefully we can have a long collaboration, research collaboration between our research group, not only in Research Center for Applied Microbiology, but because as we know in BRIN, we also have our colleague from the Research Organization for Food and Agriculture. Yeah, I think we can develop our collaboration not only with Dr. Lely or Dr. Neo team, but also with our other colleagues in BRIN who are focusing on agriculture research. Yeah, and yeah, I think that's all, everyone. Let's hear together Prof. Wong's seminar today on molecular approach for developing a sustainable plant disease management. And so please, after the meeting, after this seminar, if you want or if you are interested in collaborating more with Prof. Wong, just contact her. So and then we can do collaboration for the future. Yeah, I think that's all from me. Thank you again, Prof. Wong, for coming. And I'm really sorry because I still not able to see you in the library, but hopefully next week, early next week, hopefully, or the end of this week, I can visit you in the laboratory just to say hello. Yeah, I think that's all. Thank you very much, everyone. Enjoy the seminar. I also open this webinar. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Mr. Fathoni. Ladies and gentlemen, before we start the agenda, we will have a photo session first. So all participants, please activate your camera. And for the committee, I to take the screenshot. I will start count from first slide. 1, 2, 3. Second slide. 1, 2, 3. Thank you. Now we are going to start our discussion for today, which will be led by Dr. Neo Endra Lelana. Time is yours.
Terima kasih Bu Wardah Tuheria atas waktunya. Assalamualaikum warahmatullahi wabarakatuh. Selamat siang, good afternoon. Yang saya hormati Kepala Pusat Mikrobiologi, Pak Dr. Ahmad Fathoni, our special speaker, Profesor Wong from Universiti Putra, Malaysia. Penyelenggara seminar ini, Mas Ippo dan kawan-kawan, Bapak Ibu sekalian yang sudah menyempatkan hadir di seminar siang ini. Today I'm thrilled to be your moderator for this seminar that will be presented by Prof. Wong. with the title Molecular Approach for Developing Sustainable Plant Disease Management. Once again, thank you all for being here. In this occasion, Prof. Wong will present current strategies and the latest research that covers molecular approach related to the plant diseases. As we know that agriculture sector has economic important role for our country. We have oil palm as the largest producer in the world, natural rubber as second largest producer, and also we have rice, maize, sugarcane, cassava, banana that among the top 10 producer, and other agriculture commodities. And forestry, we have cassia and eucalypt as the main raw material source for pulp and paper industry. And also we have sengon trees as material source for plywood. And I think all of these commodities are seriously threatened by pests and diseases. And in many cases, it could cause significant economic losses. For example, in rice, we face many kind disease, like Kapul said before that blast disease, Pyrifloria oryzae, Tungro virus, bacterial leaf blight, by Xanthomonas and others. And banana, we also face banana blight disease, Berlstonia, Sarium wilt, Zika toga, by Mycosphaerella, rubber tree fall disease by Colletotrichum, in rubber tree, and root disease by Ganoderma. In oil palm also serious problem now. Root rot disease and ceratosis with disease and acacia is also this species cannot be planted again in some areas, and probably this problem could be increasing in the future due to climate change and globalization. Before Prof. Wong share his her presentation, I would like to present her CV, and I would like now Professor Wong is visiting professor at the Research Center for Applied Microbiology for a month. She is now professor in Department of Plant Protection, Faculty of Agriculture, University Putra, Malaysia. She is also Deputy Director of Institute of Plantation Studies, and also the President of ASEAN BGPR Society for Sustainable Agriculture. And I think she conduct a lot of research, many kind of crop community like rice, oil palm, and also rubber trees. Before we begin this profound presentation, please ensure your microphone unmute when not speaking to minimize background noise, and we will have discussion after this presentation. You can directly raise your hand or can make a question in the chat room. And don't hesitate to make question in Bahasa also. I think it is understandable for Prof. Wong, because Malaysia and Indonesia have similar root language, I think. Professor Wong, you have maybe around 50 minutes for your presentation. Now time is yours, please.
Okay, thank you, Dr. Neo. Can you hear me?
Yes, I can hear you, Prof.
Okay. Okay, good. Good. Is that okay? To uh, all
I cannot hear Prof. Wong first. Maybe you wait for a minute. I think Prof. Has a little bit technical problem, and we wait a minute.
Mungkin bisa dibantu tethering dulu Pak Neo.
Ya Pak, ini sedang ini kan Pak untuk tethering, untuk koneksinya mungkin agak kendala juga di CWS ini.
Okay. Good afternoon. I'm sorry for the technical problem, disconnection just now. Dr. Fathoni, very happy to see you. I repeat again. Okay. A very good afternoon to everyone. Dr. Fathoni, Dr. Neo, which is my host, researcher, Dr. Laili, and my fellow researcher at BRIN. First of all, I would like to thank Dr. Fathoni for inviting me to participate in this visiting researcher program, and also thank BRIN, or the government of Indonesia, for sponsoring this trip. Selamat siang, Okay. Okay, I will start my presentation. Display. Is it coming? Press this. Okay.
Yes.
Okay. Okay. So as mentioned by Dr. Neo, the title of my presentation is Molecular Approaches for Developing Sustainable Plant Disease Management. Actually, I have prepared many slides, so I hope I can share. I'm very excited to share my experience, and also to establish collaboration with researchers at BRIN, and I will learn from all of you as well. So I'm attached to Department of Plant Protection, Faculty of Agriculture, and also Institute of Plantation Studies. It's not moving. Oh, okay. It's slow. Sorry. Okay. Just a quick introduction about UPM. If you have not heard about UPM, Universiti Putra Malaysia, we also say selamat datang. Our motto is berilmu berbakti, with knowledge we serve, and the right picture shows the main entrance to UPM. We have two campuses, one in Serdang, which is in Selangor State, and the other one in Sarawak State, which is near Indonesia, Kalimantan. In UPM Serdang, we have a big area, 3,000 hectares, and in Bintulu, we have about 700 hectares. A quick facts about Universiti Putra Malaysia. It's founded in 1931, which is the setup of the College of Agriculture, and then it is known internationally as one of the distinguished universities in the region. Then in 2006, accorded the status of research university, and in 2010, we were awarded the self-accreditation status, which means we can accreditate our academic program by ourselves. And 2012, we were awarded as autonomy university. We consist of about 6,005 staff, academic staff of around 1,007, and non-academic staff around 4,008. And we have 84 centers or responsibility centers, such as faculty centers, institute, as listed here. Students about 30,000, 16,000 plus is undergraduate, and 12,000 plus is postgraduates. We also have student mobility programs. So I welcome BRIN researchers, if you are interested, to come to UPM to further your studies. Okay, just a brief background on my research expertise. I'm actually focusing on plant immunity and sustainable crop protection. These are the scope. Mostly plant microbe interaction and rapid detection. Induced resistance, I work on signaling pathway, elicitors, plant activators, microbes, chitosan, nutrients, and pyraclostrobin. Host defense on phenylpropanoid pathway, RNA interference, ribosome inactivating proteins, nitric oxide, PR proteins, defensin. And then the other side of the pathogen, virulent factors such as non-ribosomal peptides and siderophores. So for rapid disease diagnosis, also I work on PCR-based DNA macroarray. DNA macroarray is actually an old method, but those who have no sophisticated machines, it can be done for screening or detection, mass detection of pathogen. Of course, qPCR and LAM. LAM stands for loop mediated amplification. I also work on nature-based biopesticides such as botanicals, biocontrol agents, RNA molecules, system biology, such as detection of transcripts, and also gene expression and metabolomics. In terms of genetic transformation, I work on agrobacterium-mediated, PEG-mediated, and also CRISPR-mediated genetic transformation. Okay, so when we talk about sustainable plant disease management, we have to talk about plant health because we are dealing with plant health. Just a reminder, in 2020, the United Nations has declared the year as the International Year of Plant Health, because it is expected to increase awareness about the importance of healthy plants in achieving the sustainable development goals. As we know, healthy plants are the foundation of ecosystem functions and food security. Sorry, I have to move this. Okay, plant pests and diseases damage crops, reducing the availability of food and increasing its cost. Sustaining plant health protects the environment, forests, and biodiversity from plant pests, addresses the effects of climate change, and supports efforts to end hunger, malnutrition, and poverty, according to the United Nations General Assembly in 2018. So here are at least 5 of the SDGs could be achieved by maintaining plant health. SDG 1, no poverty. SDG 2, zero hunger. SDG 8, decent work and economic growth. SDG 13, climate action. SDG life on land means maintaining biodiversity. And of course, there are others as well. Okay, referring to food security also is important to maintain plant health. As we know that by 2050, we will be more than 9 billion humans on Earth. I think Indonesia also has contribute to these statistics, China and Indonesia and India. And we will need to produce 60% more food. According to WHO, the World Food Summit, food security is defined as when all people at all times have access to sufficient, safe, nutritious food to maintain a healthy and active life. So it's not just sufficient food, but it has to be safe, has to be nutritious to maintain a healthy life. So as we know that plant doesn't just provide food, but also shelter, feed, fiber, and medicine. Okay, what is sustainable plant disease management? It refers to strategies and practices that control plant diseases while minimizing negative environmental impacts, promoting agricultural productivity, and ensuring long-term sustainability. So the goal is to reduce reliance on harmful chemical pesticides, preserve biodiversity, improve soil health, maintain a balance between agricultural needs and ecosystem protection. And the benefits are environmental protection, as it reduces the overuse of chemicals, long-term crop health to produce more robust plants, and economic viability because we will reduce the usage of pesticide, therefore the cost, and resilience to climate change due to sustainable practices. It will help the plants to become resilient climate change. Okay, molecular approaches refers to using modern biotechnological and molecular biology techniques to control plant diseases while minimizing environmental harm and maintaining agricultural productivity. Here are some key strategies. So maybe there are many, but I list 8. Genetic resistance through marker-assisted breeding. Second one, transgenic approaches or genetic engineering. Third one, RNA interference, RNI, which I will focus my talk on this after this. Biological control through molecular techniques, pathogen detection and early warning systems, induced resistance and priming mechanisms, sustainable pesticides and natural compounds, and genetic selection. Those topics that I put asterisks are those that I have researched on those topics. For example, genetic resistance, marker-assisted selection, pyramiding resistance genes, meaning to compile all the resistant genes in one single hybrid or genotype. Transgenic approaches could be direct transformation using agrobacterium or using CRISPR-Cas gene editing. RNAi could be, there are three types, host-induced gene silencing, which is HIGS, or virus-induced gene silencing, VIGS, or spray-induced gene silencing, which I'm focusing now. For biological control through molecular techniques, is that we need to study the plant-microbe interactions and to identify those microbes that are beneficial through genomic approaches such as metagenomics, pathogen detection, rapid detection, using molecular diagnostics, and also high throughput screening. Induced resistance is using biostimulants as primer or priming plant defenses. It could be biostimulants, could be microbes or biochemicals such as salicylic acid, or oligosaccharides such as chitin or chitosan, something compounds like that. Sustainable pesticide and natural compounds. This is also very interesting to produce biopesticides using natural bioactive compounds. I use plant extracts which contain many bioactive compounds which are antimicrobial. For genomic selection, could be using genome data to predict disease resistance, which will cut short the time of testing. Okay, the benefits will be, if we use molecular approach, it will reduce pesticide use, enhance yield and quality, and also maintain sustainability. Okay. Induced resistance is also a very key strategy for sustainable plant disease management, because defense mechanisms are activated by plant infection, stress, or chemical treatment. There are three stages actually in the induced resistance pathway. First, pathogen recognition. It could be due to effector proteins or due to elicitor proteins. These are the two projects that I'm also working on. Second stage is the signaling. Once the plant recognizes the pathogen, it will activate signaling pathways. There are two types. Systemic acquired resistance, usually initiated by the pathogen infection. The other one is induced systemic resistance, usually initiated by beneficial microbes. Third stage, these defense responses, which I will explain more in the next slide, meaning the genes that are involved, the responses. Okay, pathogen recognition. Plant employs numerous immune receptors located on the cell surfaces and intracellular spaces to perceive a variety of immunogenic signals associated with plant pathogen infections. There are two types of plant immune receptors. Those on the cell surface, and the other one is in the cell. So on the cell surface, such as receptor-like proteins, RLP, or receptor kinases, RKs, these receptors also exist in animal cells. Then the next step is the microbe-associated molecular patterns, or MEMS, or damage-associated molecular patterns, DAMs. Further, we'll continue to the pattern recognition receptors. That means they can recognize either pathogen or beneficial microbes, and then trigger the pathogen-associated molecular patterns, PAM, triggered immunity, PTI, and then activation of defense. Inside the cell, there is receptor which is nucleotide binding leucine rich repeat NR and LR, which is also exists in animals. And then this is triggered by pathogen effector proteins. It will induce effector triggered susceptibility or effector triggered immunity, and then activation of defense. So at this stage of MAMS or DAMS, the plants can recognize whether the microorganisms that approach the plant is a friend or foe or enemy. So if the plant detected MAM, which is the microbe-associated molecular patterns, it will consider it as a friend. It will activate a different system, for example, ISR, induced systemic resistance. And if it is detected the microbe as enemy or pathogen, it will activate damage-associated molecular patterns, and it will go through the PTI pathway and activation of defense. Some defense responses reported massive production of reactive oxygen species, ROS, and nitric oxide, which is also my PhD work. Also calcium influxes and activation of kinase cascades, defense genes expression, PR proteins, lignin, which is the phenylpropionate pathway, resistant genes, R genes, or synthesis of defense hormones such as jasmonic acid, salicylic acid, things like that, and also ribosome inactivating proteins also induced, which is also one of my projects. Okay, what are immunogenic signals? As I mentioned just now, effector proteins, I work on Ganoderma interaction with oil palm. We have identified some non-ribosomal peptides. And elicitor proteins also serve as immunogenic signals. I work on elicitor proteins from Ganoderma. Ganoderma is affecting oil palm. Rigitoporous microporous is affecting rubber, causing white rot disease. And these proteins are called ceratoplatinine proteins. I will not explain more on this. Maybe you can read my publication. Okay, this is on non-ribosomal peptide synthetase in Ganoderma, which is effectors. It affects or it generate pathogenicity. This is the elicitor protein. This is the protein ceratoplatinine, left side on Ganoderma, right side on Rigitoporous. Okay, host defense genes that I work on. For sustainable plant disease management, we also need to look at the resistant genes, which is genetic modifications, which is highly desirable for the control of basal stem rot of oil palm, because this disease is, we still have not found an effective control on this, so we have to use genetic methods. So understanding the defense mechanisms during the interactions between pathogen and its host is essential for development of efficient control strategies and also identification of biomarkers for early detection. So these are the 4 groups of host defense genes that I work on. Nitric oxide associated genes, which is the gene producing associated with the production of nitric oxide, which is a very important signaling molecule in the physiological processes, including defense mechanism. And ribosome inactivating protein, this is involved in defense, and this mechanism is to inhibit protein synthesis. Phenylpropionate pathway genes such as PAL, CAD, peroxidase, lignin synthesis, and defense-related genes related to cell wall, related proteins, transcription factors, jasmonic acid signaling genes, and disease resistance protein. But I will not explain more on this. Okay, just a general conclusions from the studies on the gene expression or gene defense genes. Ceratoplatinine transcripts of oil palm and rubber were identified and characterized. Signaling genes, antifungal genes, phenylpropanoid pathway genes, cell wall-related genes, transcription factor genes, jasmonic acid responsive genes, and disease-resistant genes from oil palm were identified and characterized. And these candidate genes could be used as biomarkers for oil palm defense mechanism in the interaction. That means if they are regulated, we will know that there is an infection in that pathosystem. They could also be used for development of Ganoderma-resistant oil palm genotypes in breeding program. It means we can use these potential candidates to improve resistance in oil palm. This is the case in oil palm. And these findings also could be adapted into the development of efficient vessel stem route management strategies. For example, the CP protein, RIP proteins can be developed as biofungicides as application like spray or drench. Okay, the way forward, we will go into biotech for plant protection. There are two current technology, RNA interference and also CRISPR technology. RNA interference is actually a natural cellular process used by most eukaryotes for developmental gene regulation and defense responses. And this is just a pathway to for gene silencing. Okay, so this work has been given a Nobel Prize in 2006. For CRISPR, it is also a natural system. It's called clustered regularly interspace short palindromic repeats found in bacteria for self-defense against bacteriophages, the virus. And it is a powerful tool for genome editing. Also received Nobel Prize for this kind of work. Okay, for myself, a few projects I have been doing on RNAi. The first two are more on proof of concept. That means it's fundamentals, just to see if RNA is workable, can silence the target gene or not. And they found that they are working at different degrees. The first one is on RNA-mediated gene silencing of lanosterol 14-alpha demethylase or ERG11 encoding gene in Ganoderma poniense. This gene is very important because it controls the cell wall of a fungus. So many chemicals that has been produced are targeting this target gene. Okay, to suppress this target gene. And the second one is elucidating dicer-like endoribonuclease disruption on Ganoderma poniense using double-stranded RNA molecules. This is also to show the proof of concept that using double-stranded RNA, we could disrupt the dicer-like endoribonuclease. This gene is important because it's involved in the gene silencing pathway that produce more RNA, which is a virulence factor for any pathogen. Okay, the third one is on Fusarium, similar target gene, dicer-like. And the fourth one is on rice. Also, fundamental studies just to show that to develop the protocol and also to identify the effective sequence, the double-stranded RNA that could suppress the target gene. Okay, these two are ongoing projects. Small RNA as a sustainable crop protection tool against rice blast pathogen. This is to continue from the previous studies, which is to formulate the double-stranded RNA molecules. Because we know that when we apply it to the field under the sunlight, UV exposure, it will degrade the double-stranded RNA. So we try to formulate it and to test the efficacy in the field. 6 is on Fusarium, similar work, but it's on Fusarium. That's why my project here attached to BRIN is on Fusarium wilt, working on Fusarium wilt. Okay, so I will focus more on specific project, one project on RNAi, which is also my project with BRIN collaborator in the lab here. So it's called the characterization of dicer-like proteins, the one that I've shown before. Okay, we use RNAi because it could down-regulates the gene expression by degrading mRNA. It triggers by small or short double-stranded RNA. It is easily degraded, and because it's a natural biological molecule. High specificity because it's very specific. Once we develop the sequence to the target gene, it will avoid avoiding off-target organism. We focus on dicer-like proteins, as I mentioned, because it's responsible for producing small RNAs from double-stranded RNA. And these small RNAs act as virulent factors of the pathogen to cause disease in the host plant, as involved in fungal growth, conidation, and pathogenicity. This is just a schematic diagram to show that it's the double-stranded RNAs at the top. And the first enzyme that is involved is dicer. That's why we focus on dicer. So it will, if we suppress or we remove dicer from functioning, it will stop the rest of the pathway, and there will be no small RNA produced as a virulent factor to cause disease. So this is this kind of work has been successfully applied in research for other ascomycete plant pathogens such as Penicillium, Collectotrichum, and Botrytis. So now I'm working on Fusarium, FOCTR4, and also rice blast. Okay, for this project, there are 3 objectives. The first one is to isolate and characterize full-length DCL transcript in FOCTR4. Second, to assess the effect of FOCTR4 DCL2 double-stranded RNA molecules on the gene expression of DCL2 and the pathogenicity of FOCTR4 under in vitro condition. And number 3, to assess the effect of FOCTR4 DCL2 double-stranded RNA molecules in the greenhouse on the host plant. Okay, for objective 1, okay, we have isolated 2 sequences, DCL1 and DCL2, similar to reported papers. They also have isolated 2, but at a different sequence length. DCL1 is about 4,007 nucleotide, DCL2 about 5,000. And we characterize the 2 sequences according to the SMART program, the software. And DCL1 contains 4 domains, and DCL5 contains 5 domains. The one that we target for silencing is the ribonuclease 3 family domain, because we thought that this is the active enzyme domain. We also further characterized and found that the secondary and tertiary structures are similar to human endoribonuclease dicer. Okay, so summary is that DCL1 has 4,005 open reading frame, encoding 1,005 amino acids. DCL2 4,003 open reading frame, encoding 1,441 amino acids. DCL1 protein consists of 4 domains and DCL2 5 domains. The crystal structure of the human endoribonuclease dicer is more similar to FOCTR4 dicers, with 100% confidence and 25% identity. Okay, second objective, so we synthesize long double-stranded RNAs, which is about 736 base pair using MegaScript kit. For the effect, we tested the effect on the transcript level, effect of the double-stranded RNA application to the gene expression of TCL2. And we found that starting from 1000 nanogram per mil until 2000, it has significant effect suppressing the gene expression. And for the duration, we found that at least starting from 72 hours, the double-stranded RNA has a significant reduction of TCL gene starting at 72 hours to 120. For effect of double-stranded RNA concentration on the growth of the mycelia, this has no significant difference from the control. I think we need to repeat this essay again. For assess effect of double-stranded RNA on spore sporulation, we found that 1000 nanogram per mil has significantly reduced the spore production, 1000 to 2000 compared to control. Summary 2, the minimum double-stranded RNA dose required for significant gene silencing of TCL2 was 1000 nanogram per mil. Relative transcript level of TCL2 was significantly reduced starting from 72 hours to 120 hours after the RNA treatment at 1000 nanogram per mil concentration. And microconidea production was significantly reduced at starting at 1000 to 2000 nanogram per mil. Objective 3, we evaluate the efficacy in the host plant. Figure A and figure B is similar actually. So we try to apply pre-treatment means before pathogen inoculation, post-treatment after pathogen inoculation, simultaneous means together we apply, we inoculate and then we apply the double-stranded RNA. The FOCTR4 just show the control. Okay, so from here we can see that the either pre-treatment, post-treatment, or simultaneous application, they have no significant difference, but all these three has significant difference from the control. That means there's no double-stranded RNA application. Okay, effect of duration. Oops. Okay. So the blue line representing double-stranded RNA treated plants, the red line, the control. So we can see that the average lesion length was significantly lower at day 5 and day 7 compared to the control. There's no significant difference at day 3. And for the transcript level, relative transcript level was significantly lower at day 5 and day 7 compared to control, similar to the lesion length. That means the reduced transcript, and also it will affect the incidence of lesion. If that is on the root, this is on the comb. Okay. So on the comb, disease severity was significantly lower at day 7 compared to control. Okay, 14 days, there's no significant difference from control. And this is similar to the transcript. There's disease. This is the transcript also has no was significantly lower at day 7 compared to the control. And this is just a picture to show the infection. So 1A is the 7-day, 1B is 7-day control. So we can see the double-stranded RNA treated less disease symptom. 2A double-stranded RNA treated. 2B is control. So we can see some lesion here. This is less disease. Okay, lastly, stability under control, environmental condition and UV exposure. So we did some tests also to check if the condition has effect on the stability. So figure H, double-stranded RNA molecules were detected from day 0 to day 14 under the control environment. Figure I shows the double-stranded RNA molecules were detected from hour 0 to 6. 8 hours, it was not detected. So it could survive until 6 hours under UV exposure. Okay, summary 3. DCL2 double-stranded RNA molecules can be applied as pre, post, or simultaneous inoculation with pathogen. DCL2 double-stranded RNA molecules showed significant suppression on FOCTR4 7 days after application in both roots and corms. And DCL2 double-stranded RNA molecules were stable under controlled environmental condition for 14 days, while only for 6 hours under direct UV exposure. Okay, this is just a quick introduction. This is on RNA gene silencing on Ganoderma ERG11 gene. So we have isolated the full length of the gene, and we produce a viable protoplast to do the transformation. So this is protoplast, a PEG mediated transformation in the Ganoderma. And 3 hairpin. In this case, we use hairpin RNA, not double-stranded RNA vectors to transform Ganoderma. And we show that we have transformants that show the gene expression was down-regulated, the mycelial growth was lower, and also reduced ergosterol content compared to the wild type. I will show in the next slide. And we have 8 oil palm transformants that grow from the grow into polyembryotes. Okay, so this is the 3 transformants that shows reduction in growth, okay, compared to the control, the blue and the orange bar. Also shows reduced downregulated gene expression of ERG11, and ergosterol content also reduced compared to the other transformants and compared to control. So this just to show the proof of concept that gene RNA suppression can be done in the fungus. Project on CRISPR-Cas, I just started. Project number 1, actually I'm I'm a member, not the project leader. So we try to use CRISPR-Cas to disrupt the target gene, which is URA, orotidine phosphate decarboxylase, and also the core effector protein. The second project is on rice, developing rice plus resistance using variety using CRISPR-9 mediated simultaneous mutation of 4 salicylic acid 5-hydroxylase genes. So we try to silence 4 genes at the same time. Okay, so this is in progress. Okay, so just a conclusion, what I have mentioned just now, plants have the ability and the capability to protect themselves from pathogen invasions. This is the most sustainable strategy in the plant disease management. So from all these identified candidates, such as plant immunity inducers, this one I didn't mention here, is actually, like I mentioned, chitosan, chitin, oligosaccharides, or microbes, or salicylic acid, or the biochemicals, the plant immunity inducers, microbes, and defense genes. We have identified a few. Also based on RNA technology and CRISPR, we can use all these candidates and fundamental result findings as potential biological candidates to be adopted for plant immunity enhancement. So we don't have to use chemicals, breeding programs for disease resistance, and this approach is complementary in IPM, and provide better, safer, and sustainable crop protection. It is renewable resources. All these are natural resources, and it could be developed as crop protection products. Okay, so just acknowledge, before I end, I acknowledge the funding from Ministry of Science, Technology, and Innovation and Ministry of Higher Education Malaysia, University Research Grant, UPM, and also a private company, SIMW Plantation in Malaysia, and some graduate students who did the work. The banana project has been done by Sawindri, which is from Sri Lanka. And the gene expression done by Farhani. Actually, Farhani is from Indonesia, from Jakarta. So I'll be visiting her before I go back to Malaysia. Okay. Okay, before I end, just want to promote a conference. IKP, Institute of Plantation Studies, is organizing the 3rd International Symposium on One Health, One World, which is on the 10th to 12th December. So I invite all those who are related to the conference. And the theme is Empowering Planetary Health Through Science and Engineering Innovation, which is a very interesting topic. So if you want to have more information, please go to the website, IKP website. I also would like to invite BRIN researchers. If you are interested to study in UPM, yeah, you can contact me or Dr. Fathoni. Thank you so much. Terima kasih for your attention.
Thank you. Terima kasih, Prof. Wong, for the insightful presentation. I just realized that UPM has a very good area, yeah, around 3,000 hectare in Selangor. It is as huge as one sub-district, apa itu, satu kecamatan ya, kira-kira 3.000 hektar. Now we start for the discussion. I would like to invite audience for the discussion to dig deeper into what Professor Wong has already presented. You can directly raise your hand, or you can write in the chat room. Maybe there are a lot of topics that just Professor Wong presented. A lot of molecular techniques like transgenic RNA, pathogen diagnostic, industrial resistant, and Professor Wong worked a lot in oil palm, yeah, palm and also rice, maybe with RNA inference for down regulates gene expression.
We have some question on the chat.
Yeah, we have one question from Pak Nurfianto Giar Pangindung, ya, in chat room. 3 questions. Yes, 3 questions from Pak Nurfianto. First question: how the development plant resistant or tolerance to virus, especially in horticultural crops such as chili, tomato. And second question: what the best strategy and approach for plant disease such as potatosis nematode, Wang Longping disease, bacterial leaf blight in rice, fusarium wilt disease? Almost all plant infected with this fusarium, Ganoderma in oil palm, that until now there is no effective control yet. And the last question is: the molecular approach able to reduce, improve the incidence of plant that no longer resistant or tolerant to certain diseases? For example, in sugarcane, the bulu lawang variety was previously resistant to smooth disease, but due to excessive racun, its resistance is now broken. I think Professor Wong can directly give the response on this question. Please.
Thank you, Professor Nurfianto. Right. Thank you for the very complex question. Question 1, how to develop plant resistant tolerance to virus. Okay, usually virus is easier to develop using genetic modification, especially RNA interference. We can use Higgs host-induced gene silencing. We clone a fragment of the target gene of the virus and we transform it into the host. Then the RNA interference pathway will be activated if the virus infects the plant again. This is the transgenic plant. So this has been successfully used for other disease resistance against virus. So they usually use this transgenic. So the thing is whether the country or whoever consumer is accepting GMO plant. Okay, but I think I know of many countries they have transgenic eggplants, transgenic corn, all those things. Eggplant especially because we eat them from Bangladesh and also Pakistan. They mentioned that they have to use transgenic eggplant. Terung, we call terung in Malaysia. So sama, okay. So yeah, so they said they have to use transgenic terung so that they can eat terung. Otherwise, they cannot plant because of the borer, in borer. So for virus disease resistance is usually a bit easier to generate GMO. Other than that, if you want non-GMO, it could be cross protection. You can apply RNA molecules, like I mentioned just now, double-stranded RNA can spray on it. Yeah, it can be. You can try that. The second question, best strategy to approach plant disease such as potato. Okay, nematode usually is crop rotation because nematode is the juvenile too is very specific, host specific. They need to find a host. If they can't find the host, then they will die. So crop rotation or intercropping to break the cycle of the nematode, that is a very effective approach. Panglongping is caused by vector, so we have to control the vector. Bacterial leaf blight, that is bacterial leaf blight usually copper-based chemical the farmer use. The other alternative is biocontrol agent. Okay, we have found some biocontrol agent to control bacterial leaf blight. So but there's also a new variety of rice. Like in Malaysia, we have a new variety of rice that is more resistant against bacterial leaf blight. Fusarium wilt, it's very difficult to control. That's why I'm working on this plant in BRIN. Yeah, we use microbial and also nutrient control is more effective. Ganoderma is a big problem. That's why Ganoderma usually we try to clear the land, clearing, and to apply microbe just to control the soil with good microbial development population. That could help. But the problem is with the plantation, the company, they do not. They just want to use easy way, just chemical fertilizer, chemical pesticide. So they don't really take care of the soil. That's why it's difficult to control Ganoderma because the soil has not many variety, no diversity to control Ganoderma. Third question, molecular approach to reduce incidence of plant no longer resistant or tolerant to certain. That means they overcome the resistance. Yeah, that's right. This is very common because pathogen and plant, they are like tug of war. Who is in the forefront, they will win, right? So usually resistant variety, it can be broken. So we try to complement with if farmers still use chemicals, they have to use a different active ingredient, not the same active ingredient the same time, so they have to rotate. If they using microbe is the best one, but it's slow. In the soil, in the soil to keep the soil microbial diversity healthy. Okay, that is one way. Sugarcane, I have no experience in sugarcane, but yeah, if smart disease is Basidium mycete, yeah. So it's difficult, like Ganoderma and Rigitoporus, they are Basidium mycete. So they have the Basidio cup, so that's why it's a bit difficult to control. So usually it's more on cultural microbe, maintain the soil health and reduce chemical use that will improve the microbe diversity better, then it will help the plant to thrive, to be strong, and to resist the disease. Okay, thank you. I hope I answer your question.
Thank you, Professor Wong. I hope the answer is satisfied for Pak Nurfianto. While we are waiting, someone raise hand. Maybe I should ask Professor Wong.
Biological control.
Oh yeah, Pak
One more
Nurfianto ya.
question.
A question from Bu Nita Sari.
Endra,
Yeah, could you explore a little bit more about the study of biological control through molecular technique, especially plant microbe interaction, because I have also doing that kind of project. Please, Professor Wong, respond this question.
molecular technique in regards to biological control just mean that you have to identify what are the diversity, the interaction between the host plant and the microbe. Identify what species are beneficial to the host, and then we identify through molecular techniques and also the population as well. They have different group functions, so we identify what group are there. That's using molecular technique. Other than that, microinteraction, yeah. So molecular technique is just to use for detection of the species, detection of what kind of metabolites they produce, probably what kind of what genes they induce in the plant. These are the plant micro interactions using molecular techniques that we can use. Transcriptomics kind of too. Yep. It can be transcriptomic, can be genomic, can be metabolomics. Okay. Yes. That was a lot of information there. Yeah. Thank you.
Thank you, Professor Wong. But maybe has satisfied for Mr. Professor Wong response. Professor Wong, I have a question that you study RNAi and CRISPR. Is it categorized as GMO or not?
Very interesting question. Many people ask about this. Okay, as I mentioned, RNAi, there are three types. One is host-induced gene silencing. One is virus-induced gene silencing. One is spray-induced. Host-induced means you transform the plant. So that is GMO. Virus-induced, I think it's the same thing. You transform the plant with the virus fragment. Spray-induced is non-GMO. We just spray the molecule and then it will work like activate the plant RNA system, the pathway. And CRISPR is editing the genome without inserting any extra DNA. Many countries accept it, like US, China, but European not accepting it. So it depends on the country. So yeah, so in your country, if your country accept it, then should be okay. You can do the research. Yeah, I hope I answer your question.
Yes, already answered the question. I'm
No more
still
question.
waiting. Maybe while waiting, someone raise hand. I have one question more. Yeah, we in forestry we face a big problem ya in acacia mangum with Ganoderma bilipi, root rot disease, and ceratocystis disease. In some area, acacia cannot be planted again because loss is more than 50%. Yeah, maybe Prof has suggestion to overcome this problem. I mean that how we can plant the acacia again next in the future, because now it should be replaced by other species. Yeah, currently many plantation plant eucalyptus to replace acacia. Maybe Professor Wong have some suggestion for the next future that acacia can be planted again in this
Okay.
area.
All right. Thank you for the question. So this similar Ganoderma acacia is philippi, right? Ganoderma philippi. It is similar to an oil palm Ganoderma. I mean, the case is similar to oil palm infected with Ganoderma boninense. Yeah, it's a very difficult disease to control. Once it's in the soil, it will remain there if you do not clean it when you replant the next season. So what the oil palm plantation did, usually they will clean the land, remove all the debris, and expose it under the sun for quite some time. That's one way. And using microbe, okay, applied microbe before transplanting. Maybe in the seedling, you put microbe such as mycorrhiza, trichoderma, your original natural species, and to strengthen the root system before you transplant it into the new area that you're going to plant. And of course, the history also very important. Maybe you don't use, if you have choice, you don't plant in the same area, maybe other areas. If you have no choice, then you have to do clearing, and also apply microbes. So I think the soil health is very important. It will take care of the pathogen, actually, the beneficial microbe will take care of the pathogen. So, and also to make the plant more vigorous and healthy. Those are the main thing. But if it is infected already and you want to control the spread, then people will use chemicals. That will be the fastest way. Other than that, prevention is much better. Thank you.
Okay,
Agung has
thank
a
you,
question.
Prof. Wong. Yeah, I think there is one question in the chat room from Pak Agung. I may have missed the explanation or NNI research. I would like to ask, how do you ensure that this applied double-stranded RNA is stable? Is the double-stranded RNA applied once or several times?
Okay, thank you for the question. So from the banana case that I presented just now, we are trying to find out what is the timeline that is stable. For now, we applied once and we check the duration. So mentioned that is after 7 days. I mean, until 7 days. That's why the new project, we would like to formulate the double-stranded RNA so that it will be more stable, especially under the UV in the field. So we will try to formulate it and make it stable. All right, thank you.
Profesor Wong,
Ada lagi satu. Lagi.
Still from Pak Nurfianto. I want to ask again for spraying RNA molecules to plant. How the development of this type product like synthesis and production, formulation, packaging, cell life, how the comparison with other biocontrol? Thank you.
Okay, thank you for the question. Yeah, so my project now is at the proof of concept level. So the next step is to mass produce it. Once we have identified the efficient or effective molecules, we will mass produce it. And we found that using the kit is very expensive for mass production. So we read in the publication there are a protocol using bacteria to mass produce the small RNA. So we have to construct vector with the gene that produce that one way for mass production. Formulation, yes, we are trying to do the formulation. Also, there's some research done on the formulation, what type of formulation. So we are trying to optimize that now in the current project. Packaging, shelf life, yeah. So actually for RNAi project, RNAi products, it has been used quite successfully as reported for the insect control. Okay, they have product of RNAi to control insects. For insects, is a bit easier because they digest the RNA, they just uptake the RNA molecule directly. So that is effective, very effective. But for diseases, it's a bit difficult because we have to apply it to where the pathogens are. So that's why it's a bit difficult. We need formulation and a long shelf life. So formulation will improve the shelf life. So that one, we are working on it. I think in publication elsewhere, you can find those information related to the insect control. Actually, they have products commercialized for insect control. Okay, how comparison with other biocontrol? So this is, as I mentioned, we can try to compare the insect products, the RNAi product that control insect. As I mentioned, to control disease is a bit complex. We have to do more research on that. Thank you.
Thank you, Prof. Wong. And there are several questions again in the chat room from Bu Icha. How long do you think to do the research related to get the resistance plan of a pathogen? And how about the consortium or combination among the kind of microbes?
Okay, thank you. Let me read the question again. How long do you think to get resistant plan of pathogen? How long? Okay. To produce a resistant plant variety is very long, but conventionally is long. But with biotech, I think it takes less than 5 years for biotech. That means you transgenic do transgenic. Consortium, yes, is a very good idea to use consortium of a combination of microbes. Preferably they are compatible, preferably they have different functional groups together so that it can work together and it will be a broad spectrum. Okay, thank you.
Okay, thank you, Prof. Wong. Question from Bu Nurlaili. I would like to ask about the strategy to develop an effective biocontrol formulation to suppress the plant disease, especially using bacteria consortium. May you have any suggestion or consideration for it?
Okay, thank you. Similar to the question before, consortium, yes. As I mentioned, we have to test the compatibility of the different group of functional group. Okay, not just different species, but different functional groups to be in the formulation. Yeah, that's my suggestion. Thank you.
Thank you, Prof. Wong. I think that's last question. And now time is almost 3 o'clock. I think we can finish this seminar today. Thank you all for your thoughtful question and for being such audience. And I also like to extend a special thank you to the speaker, Professor Wong Wiyun, for sharing their expertise and insight. Waktu saya kembalikan ke Bu Wardah. Terima kasih. Thank you.
Thank
Terima
you,
kasih.
Dr. Endra Lelana, for leading a very interactive discussion. And thank you, Professor Dr. Wong Wiyun, for a very interesting presentation. And as a token of our appreciation, we will give a certificate for Professor Dr. Wong Wiyun as a speaker. Thank you very much for sharing your valuable knowledge.
Thank you. Thank you.
Thank you,
Ladies
Bu
and
Wardah.
gentlemen, we have finally come to the end part of this webinar. Before I close this session, I would like to ask Head of Research Center for Applied Microbiology BRIN to give us some short closing remarks. To Dr. Ahmad Fathoni, time is yours.
Thank you, Mbak Wardah. Yeah, thank you, Prof. Wong, for your really nice experiences on several approaches. Yeah, you're doing RNA and then CRISPR, and I'm also interested in kind of spray RNA. I think this is very interesting approach, yeah, because I was wondering whether we can actually design the product not only for biocontrol but also for biofertilizer or like a plant growth inducer but using spray RNA. I think this approach very interesting, everyone. So we can actually learn or use this kind of approach for another application if it is possible. Then Prof. Wong, I'm sure she will be very happy to share her experience in the laboratory, how we design this kind of experiment. And maybe also Pak Neo or Bu Leli already mentioned that actually we or BRIN have a banana project as well through BMGF Foundation. But and you also have banana project. We have banana project. I think we have very much similarity and on kind of a project where we can share on several approaches to achieve our project goal, yeah, in your side and also in our side. Yeah, again, thank you, Prof. Wong, for your kind experience that you have shared with us this afternoon. And everyone, please welcome any collaboration with Prof. Wong. Just come to the laboratory in Cibinong and then feel free to have discussion with her, and also even Prof. Wong may also invite her colleague also on similar or related topic with our here in BRIN. And also we also see that Ganoderma and then palm oil is also one of kind of big activity also in UBM, where also we have very much interest in this kind of research, and we also have research group focus on Ganoderma research as well. And then I already mentioned to to our colleague that we can also invite Ganoderma people from UPM to Indonesia, like Dr. Aswad. I know that he's doing PhD in Ganoderma palm oil, and then next year, we will have quite big project also on Ganoderma with one of big company or industry in Indonesia. I think this is this kind of scheme or through webinar or visiting research can also help us, running our project from BRIN or from UPM side. Yeah, again, thank you, Prof. Wong, and everyone for joining us and a very fruitful discussion, I think. And again, I hope, Prof. Wong, we can do some if not some big project, but some project, I think I'm sure we can do something, maybe we can develop proposal first, together, and then later on we can try to get research grant from BRIN or from overseas. Okay, thank you, and hope to see you soon in the laboratory, Prof. Wong. If not this week, hopefully next week I can see you in the laboratory. And again, everyone, thank you. I close this webinar. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi
Thank you,
wabarakatuh.
Dr.
Thank
Fathoni.
you, Mr.
Thank you,
Fathoni.
Prof. Wong, everyone. Okay, thank you, Mbak Mada.
Ladies and gentlemen, this is the end of our agenda for today. I'm Moderator Haria on behalf of the committee. I would like to say thank you very much to our speaker, moderator, and all participants. See you in the next event. Wassalamualaikum warahmatullahi wabarakatuh. Thank you.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, everyone. Selamat sukses Bapak Ibu semuanya.